Hello, everybody. Good morning. I'm Carlo Cerrano from the Polytechnic University of Marche, Ancona, Italy. And thank you very much for being here today and joining us to the 2022 webinar series, Mediterranean MPA Facing Climate Change. In today's webinar, we will continue focusing on the monitoring of the climate change effect in Mediterranean MPA. And today, we are welcoming more than 139 registered participants and coming from more than 15 countries. So this webinar is organized by the MPA Engage project, monitoring protocols to track climate change in Mediterranean marine protected area to face climate change, in association with two other projects that are MPA Networks and AMAre Plus, that are all projects funded by the Interreg Med project. The main goal of MPA Engage is to promote the resilience of Mediterranean marine ecosystem faced to climate change by developing a full toolbox for MPA managers. This toolbox is intended to be used across the Mediterranean MPAs and beyond. Now, I just introduce some few technicalities. Before we start, some general information concerning the webinar. The webinar will be recorded and posted in a YouTube channel afterwards, after the meeting, after the webinar. And during the presentation of about 1 hour, you will be able to post questions using the question box at any time at the end of the webinar. And we will dedicate around half an hour to opening the floor to the participants through answering the posted questions. We will do our best to answer directly by email those questions that will not be able to take during this session if time is not enough. And please feel free to share your impression about the webinar using the hashtag MPA webinar on Twitter. Then in this webinar, our speakers will show, I don't know if you can go ahead with the slide. Yes, thank you. Also the next, yes, thank you. In this webinar, our speaker will show how to monitor the conservation status of Posidonia oceanica meadows, Pinna nobilis, and sea urchins population, and how their status can be related to climate change effect. The training will illustrate how to perform the field survey, data upload, and information sharing. 3 protocols will be presented that are Posidonia oceanica fast assessment, called POFA, fast assessment of Pinna nobilis conservation, that is FAP, and ARC, that is the sea urchin population. The today webinar panelists are Ivan Guala, Daniele Grech, and Joachim Garabot. And I let the speaker directly introduce themselves. So please, if Ivan and then Daniele and Joachim will just say a couple of words to introduce themselves. Thank you, Ivan.
Thank you, Carlo. Thank you. Good morning, and good morning to everyone. I'm Ivan Guala. I am a field researcher associated at the IMC, the International Marine Center of Oristano in Sardinia. My research activity mainly focuses on priority macrophyte habitats and conservation of benthic systems. And within the project MPA Engage, I was in charge to develop the monitoring protocol, a simplified monitoring protocol to assess the conservation status of Posidonia meadows and to track possible change related to climate change.
And Daniele, if you want to introduce yourself briefly.
Hi, good morning to all the audience. I'm Daniele Grech, marine biologist, speaking from Sardinia. I'm a researcher at International Marine Center, and I'm mostly working on Mediterranean marine habitats and species with a special focus on conservation and spatial planning. Thank you.
Thank you. And then we have our fixed guest for this webinar, Joachim Garabot.
Good morning, everyone, and thank you all the audience and participants for joining the webinar. I'm Joachim Garabot. I'm a researcher, a marine biologist working at the Institute de Ciencias del Mar in Barcelona, Spain. And my research is focusing for the last 20 years, I would say, to track changes, the impacts of climate change in the Mediterranean. And today I'm going to present the monitoring protocol on how to track sea urchins populations. Thank you.
Great, thank you. So, well, we can start with the webinar. So, Ivan, if you want to start with the protocol on Posidonia.
Yes, okay. Thank you again, and good morning to everyone. So I'm going to present the first monitoring protocol of today. POFA is the nickname of the protocol, that is the Posidonia oceanica fast assessment of meadow conservation status. I think that all the audience know what Posidonia is and the relevance of this plant and the habitat that the plant form for the Mediterranean coastal systems. Posidonia is very important for primary production, for releasing oxygen and uptake carbon dioxide, for trapping sediment, and sediment stabilization. And then the meadows are able to provide habitat, refuge, and nursery for many species. So these meadows provide relevant and consistent service to the environment, to humans. For this reason, Posidonia meadows are included in as a priority habitat and of conservation interest in different international and agreements and directives, such as Barcelona Convention and Habitat Directive. The reason of this conservation is that this habitat is vulnerable to many human stressors. For instance, pollution, chemicals, over-sedimentation, mechanical impacts. And so this habitat is threatened by a lot of anthropogenic pressure. But in times of...
Sorry, Ivan, can you just put the presentation in full screen?
Yes, it is.
No, because we are seeing it's not in presentation mode.
Now?
No, it's not. We saw the presentation, but it's not in presentation modality.
Oh.
Try to click. Okay, try again to click this.
Yes, I do this.
But no, it doesn't change.
I don't know what's up.
Okay, anyway, for the moment, go ahead in this way. Just.
I'm sorry for that.
Okay.
So in these times of climate change we are experiencing, it's been demonstrated that the Posidonia oceanica is also sensitive, highly sensitive to warming. In particular, shoot mortality rates increases with increasing water temperature. And model approaches predicted that for future years, shoot density is expected to decline under climate change scenario due to increasing temperature. Another fact is that increasing temperature also affects the flowering of the plant. Flowering events generally are considered very occasional, but in the last year they are increasing, probably due to increasing temperature. And this can be an adaptive response of the plant to the stress caused by heat waves and increasing warming. Starting from these evidences, within MPA Engage project, we decided to develop a practical and a simple tool to support MPA managers for tracking change over time in shoot density and flowering events. The tool is addressed to MPA managers and also marine conservationists and scientists, but it is designed to be applied also by some categories, such as divers, volunteer divers, because for applying these methods, no specialized background is necessary, but just to be a good diver and to be accurate. And of course, training and intercalibration processes among divers is strongly recommended. So, as I told before, the main restriction of the protocol are shoot density, that is the number of shoots per square meter, and the presence of flower or fruits in the meadow. Shoot density is one of the most common descriptors used in maybe most of maybe all methodological approaches and indexes developed in Mediterranean for assess the conservation status of the meadows. So we decided to use this.
Sorry, Ivan,
yes.
because since we have this problem not displaying the full screen, you have to move the slides here, otherwise we cannot follow your the different slides.
Okay.
Yeah, exactly.
Sorry.
Fantastic. So if you can then move here, it's going to be, then we take advantage of the nice presentation that you did. Thank you.
Okay, thank you. Sorry for this inconvenience. Shoot densities is very commonly used, so we decided to maintain this descriptor in the protocol, because this is very informative on the status and the structure of the Posidonia meadows. Flowers occurs generally in autumn, while fruits are fully developed in spring. These events are not constant, but in the last year, precisely because of the increase in temperature, flowering events are increasing more and more. Other additional information can be collected during the protocol, the monitoring. For instance, the type of substrate. Posidonia is able to grow on sandy, on mud, and on sand, on mud, and on rocky substrates. Also, the type of the meadow can be relevant. The continuity of the meadow, if the meadows cover the 100% of the seabed, or if there are clearings due to natural reason, for instance, sandy and rocky clearings, or for human impact. The presence and the status of Pinna nobilis and other seagrasses can be also interesting to be collected, and also the presence of alien species and mucilaginous aggregate that can cause the detrimental effect to the plant, and the presence of potential pressures such as waste discharge and sign of damages on the meadow. This information can be collected at the site level. So go in the detail on the activities, which is the material we need for the protocol. If possible, a georeferenced map of the area of the MPA of the meadow, and then simple tools such as diving buoy, a GPS to mark the position of the stations, the sampling station, 50 by 50 centimeter quadrat, that is our sampling unit, a diving computer, and a plastic board to mark to record the information and the data collected. As to the georeferential map, this is a map provided by Tavolara MPA, that is a partner of MPA Engage project. They divide all the surface of the MPA in cells 100 by 100 meters. This can be useful to design, to plan the sampling design for the monitoring. Each cell has a cell code and a georeferential position. That is the place where we have to go for perform the monitoring. Once we are in the center of the cell, we can drop the buoy, and the divers go down to do the monitoring. They have to count the number of shoots within 9 quadrats, if possible divided in 3 groups of 3 quadrats, in a radius of 50 meters from the center of the cell, from the diving buoy. And to do this, once on the middle, the quadrant is pushed on the bottom in order to count every single shoots. One shoot is composed by a rhizome and some some leaves, and all the shoots have to be counted within the quadrats. Within the quadrat, we can also find flowers, and so we have to count also the flowers within the quadrats, then record the depth of each quadrat, the substrate type, and then when we move to another, to the next quadrats, we can visually record the presence of flowers outside the quadrats. Then we can record the data, the information collected in the field in this plastic board. If possible, you can pre-organize your plastic boards in data fields, and record all the information collected for each quadrant for the site, and there is also a semi-quantitative scale for the presence of Pinna nobilis and alien species such as Caulerpa racemosa, Cilindracea, and Caulerpa taxifolia. This plastic board is provided as annex to the protocol in the MPA Engage web page. The best period for monitoring is the end of summer or the beginning of autumn, because in that period the leaves are shorter, and so it is easier to count the shoots in the quadrats, and also because in that period in autumn you can find more likely the flowers. As to the sampling sites, the number of samples depend, of course, on the local condition. In very huge MPAs such as Tavolara, it's possible to select more sites. In any case, at least 3 sites should be selected at each depth. In other cases, depending upon the size of the meadows, the number of sites can be smaller. For instance, in Briuni, that is a very small Briuni MPA, a very small meadows, only 4 sites can be selected because there is no large Posidonia meadows. Okay, once collected this data, we have to manage them, and the first thing is to fill in the data in the enter the data in the database. The protocol provides a data file with different data sheets. Some of them are guidelines to fill in properly the other sheets. But the more important data sheets are this one that is that collect where you enter the data at site level. And then this one when for entering the data at quadrant level. Each row is a quadrat, is a replicate, and for each replicate you have to enter the generic information about the name of the location, the GPS position, and then the relevant data, that is the depth of each replicate, the number of shoots in the quadrat, and the number of flowers you find in the quadrat. Then the file generates automatically 3 information: the depth range, shallow, from 0 to 20 meters, intermediate from 20 to 30 meters, and deep, lower than 30. Then the number of shoots per square meter by multiplying the number of shoots in the quadrat and then the flowering index, the intensity, that is the ratio between the number of flowers in the quadrats and the number of shoots in that quadrat. As for shoot density, there is a interpretation scale provided by Sparac that give information about the ecological status of the meadows, depending of the number of shoots, of course, and the depth. So you can find the ecological status, and then you can also assess the level of impact for each condition. This interpretation is provided as annex to the protocol. As to flowering, in addition to flowering intensity, you can also calculate the flowering prevalence, that is the number of sites of middle flowering in a given year on the total number of sites monitored at each MPA. These descriptions, the density, shoot density, and flowering events have to be used with caution, because you cannot provide to be sure about the impact assessment with only one observation. What is important, and this is a part of the philosophy of MPA Engage, is to collect the data over long time series. Only with the long time series you are get information and to disentangle the effect of climate change and warming to other to the effect of other anthropogenic pressures. Results within the project are useful tools to show the results and to help managers in identifying the most concerning situation is has been developed by Power BI. And this is just an example related to the Posidonia oceanica meadows in Sinis MPA in Sardinia. 3 years of monitoring in 3 sites, and the Power BI provided the data, or the average data, in the upper part of the slide, and the assessment of the impact in the below. And for each sites, you can assess the trend of the shoot density and the consequent level of impact. Just some final remarks. MPAs are key players for the protection of the sea and the early detection of change in environmental conditions, but often resources are not enough to meet all the management needs. And moreover, institutional monitoring programs often are discontinuous in time and space. So POFA protocol is simple, is time and cost effective, and so we think this is it is suitable for annual data collection over long time periods. That is the need to have useful information about the evolution of the meadows. Long-term data collection are crucial for understanding the causal variability and how much climate change affects this variability. Finally, POFA protocol can integrate institutional monitoring, because shoot densities is common to most of institutional monitoring, and coupled to the assessment of main local pressure, can contribute to unravel the effect of different impact, and then support manager in identify action for conservation of this habitat. So that's all for my side, and thank you for your attention, and I leave the floor to the next speaker. Thank you.
Thank you, Ivan. Very clear presentation, a lot of information, and I hope we can discuss later this protocol. Now we can leave the floor directly to Daniele for to introduce the methodology regarding the assessment of Pinna nobilis. Please, Daniele.
Yeah, thank you, Carlo, for introducing me. And today, as explained before, I'm going to introduce you how to monitor Pinna nobilis conservation and health status. But before to go into the deep, I would like to share with you the video biopills that that regards the target species of this protocol.
At the bottom of the sea lie hundreds of dead fan shells, petrified stalks, motionless monuments to lost vitality and beauty, enormous bivalves, known since the ancient Greeks and icons of the Mediterranean Sea. A rough shell, internally shiny with iridescent mother of pearl. A new deadly enemy is leading them to the brink of extinction. A microscopic protozoan is decimating the whole population. The epidemic has struck violently. Since 2016, no country has been spared from the disease. The occasional discovery of living individuals may rekindle hope that the species can survive extinction. Monitoring, protecting, and reproducing healthy individuals is our only hope to save the noble pen shell.
Thank you. This video was developed in the framework of the MPA Engage project to spread the information on this important ecosystem engineer species. But now we can go directly to the presentation to go into the deep of this protocol. As previously anticipated, the target species of this protocol is the noble pen shell Pinna nobilis, but eventually also another cogeneric species, that is Pinna rudis, that you probably knows. So the primary objective is just to determine the abundance, the site structure, and the health status of Pinna nobilis population within the MPAs. Pinna nobilis is important. Why? We saw in the video. It's considered an ecosystem engineer. It means that provide an habitat for many other species. It's can be considered also an habitat a species that add another habitat, for example, for soft substrates. So increase the heterogeneity and increase the biodiversity. It's a substrate for many other species. It's ecologically important because it has a filter feeder function. The high volume of water is filtered, and these allow to also to decrease the turbidity. So the ecosystem services that provide are related to the ecosystem functioning, but also to the other let's say other important functionalities for the ecology of marine ecosystems. It's a long-living species, lives up to 15 years and more than 1 meter or more than 1 meter high. And for these reasons, it can be considered a sentinel species. As you know, is currently struck by a disease at the entire Mediterranean scales with unprecedented in an unprecedented situation never reported before. Many papers came out from 2016 reporting the mortality all around the Mediterranean, like waves from the western side to the eastern one. What is the problem? Probably many of these of the parasite have attached the species, and this is a multi stress pathogenic illness. This is very important, Pinna nobilis, especially because there is a long historical tradition in the Mediterranean Sea. It was exploited also locally as a food or for gods, like they were precious fabric, like the sea silk. In the Mediterranean, there were many masters of sea silk. One of them still he's operating in the southwest of Sardinia, Chiaravigo, And here I report many of this fabric, precious fabric that have been done from the past. So also a very important cultural, let's say, important for this species. Here you can say the material that we can use to apply the protocol. They are quite easy to apply the protocol, and you just need a GPS, underwater compass, some buoys to mark the position, a rule or frame with subquadrats, and a plastic sheet with the protocols to mark all the numbers for the monitoring, and of course pencil and possibly an underwater camera to shoot some photos in case of doubtful specimens or the monitoring by measure on lab. The requested skills are that you have a diving certification and you are capable to conduct a monitoring underwater, to be accurate and with scrupulosity to search the individuals that not often are easy to detect, especially for the young one, the recruits. We will talk with this later. And of course, you have not to be specialized as researcher, but trained, and intercalibration process, of course, are needed. This is the protocol that is inspired from the previous one from ISPRA. Basically, you have to do a belt transect, 100-meter long, and with a width of 3 on the right and 3 meters on the other side, so 6 meter. And in this way, you will count all the individuals that are the living one on this surface. You have to mark the geographic coordinates of the beginning and at the end of the transect, and in the bottom, of course, you have to go straight with a constant direction using a submersible compass. An important thing is that you have to be careful considering the specimen. Probably at this moment in the Mediterranean, we will if we will find some alive specimens, we are lucky. But the protocol developed in MPA Engage project is perceived has been created to have a long life and to considering also that will be applied hopefully when the population will rise once more. So you have to consider to check for the dead specimen that are standing without the mantle inside, the dead dropped, or the diseased that have a slow closure or not completely closing when you approach to the individual, and the hopefully alive that have a fast closure, so the parasite didn't attack the specimen. So we are interested in these two, the diseased and the alive. In case you have many alive individuals, you can also did a photo with a reference with a scale. Otherwise, into the floor, you can see the alive specimen and mark the unburied length, the maximum length, the maximum width, and the minimum width. These are the three most important measures that you have to take of an alive or a diseased individual. It's important to stress that in some cases, the two species of Pinna nobilis and Pinna rudis, can share the same habitat. Recent papers came out on this issue, and it's important to stress to say that Pinna rudis seems to be not affected by these mass mortality events. So the fast assessment protocol for Pinna nobilis offer a rapid tool to census also the Pinna rudis. Here are some characteristics difference in morphological characteristics in which you can distinguish the two species. The Pinna rudis has large and widely spaced spines that are well marked even when adults. Spines are also present in Pinna nobilis, especially in the juveniles, but they are small, distorted, and very close to each other. Has been reported also the potentially of the existing hybrids, and that's why it's important in case of doubtful specimen to take some photos to be sure which species can be and to go further inside and research on in these individuals. So you should treat the Pinna rudis specimens like a Pinna nobilis in the data sheet, and then of course in the notes to put that the species is belongs to Pinna rudis. The monitoring of the Pinna nobilis protocol, of this protocol in general, of the pinnas, will provide the density, the percentage of affected individuals, the percentage, of course, of mortality, the size structure of alive individuals and of former population. This is our underwater board to mark all the measures of the live specimen and the transect. For each transect, you have to take note of the depth and the coordinates, the temperature of the water, and the name of the server, of course. And then you have many rows with all the individuals that correspond to each individuals, where you can put the three most important measure to assess all the individual. Let's go to see the Excel data file to provide the information. These tools are all available in the web page of our project MPA Engage and can be downloadable from that pages. When you open the data file, you can see clearly all the guidelines to fill all the Excel sheets, and also the different level of information. We have the site level, the transect level, and the individual level. We will see then how it works. Here you can edit all the information with the depth, the alive and affected individuals, and also all the other information regarding the habitat and the dead individuals that of course have not to be measured but have to be considered inside the transit. What's about the period of monitoring? The best one is late summer, early autumn, not because of the characteristics of the biology of the pinna, but because of the phenology of one of the habitats where it's most spread, the pinna, that is Posidonia oceanica. Of course, as also commented by Ivan, in this period, Posidonia oceanica leaves are shorter, so this will allow you an easier and careful, more careful, more clear specimen identification, especially for the little one. Since in this period we have some, let's say, we have some evidence that recruits are some recruits are still happening. So probably you will have chance to find the little one that are trying to naturally recover, and of course, especially in Posidonia oceanica, but also in little boulders, they are not easy to be identified. So also the coverage of macroalgae is low at shallow depth, so we'll have more chance in this period. And the sampling size, concerning the sampling size, we suggest to at least choose 3 study sites per MPA, and the suite of habitats, it depends on the MPA, but should be seagrass beds, detritic bottom, and rocky boulders. This is a specific tool that was developed in the framework of MPA Engage that allow to visualize, synthesize, and display our result, our preliminary result. This is an example of our marine protected area where you can see the results at different levels. Here you can see our protocol here, and at the MPA level information, you are visualizing the percentage of dead individuals. Here you can see in the menu. Here you have undamaged, juveniles, and 2 levels of information. And here you can see the assessment with all the years and the different deep intervals. Here you can see the undamaged ones, and you have all the statistics, the assessment, if the Pinna that was old, very old, recent, or very recent. And here you can see the juveniles, for example, the percentage of dead specimens per site. This will be automatically provided by this powerful tool that allow to synthesize the data that you are sending us. Here an example of related to this site, and here another one. So you can see the percentage of dead specimens, and here you can see also the three depth range: shallow, intermediate, and deep range, considering the reported depth. Another important thing to stress by approaching to the end of this presentation is that the good news that Europe recently financed 2 important life projects focused exactly on this species, Pinna nobilis. So there is evidence of little natural recovery, but of course the species will be helped probably, hopefully, to the restocking of the species, since the mortality at the moment is really very high. And we need, of course, synergies and data share with scientific community to try to restore, to try to this type of habitat, and this species. I would just to stress on few important points concerning the protocol, and in general also the framework of the project, that MPAs plays, of course, a key role for the detection and mapping of this climate change related species, and in particular the disease and the survival spot of Pinna nobilis. This protocol, as you can see, is simple, not time consuming, not money consuming. So just apply it and spread the protocol, and because it is applicable to large scale monitoring and baseline building. So you have to apply large scale monitoring in sensitive habitat, and this of course should be prioritized, especially in the habitat like for example the paralic environments, that seems that for some of the species represent a place where still there are some living individuals. And at the moment the most important thing is to detect and to find the alive specimen that still exist. So you have also to take into consideration citizen science to fill this lack of knowledge across MPAs. But in general, at the moment, the important is to share with toolbox, with toolkit across MPAs and Natura 2000 network. Okay, I finished. Don't forget to leave a comment in the chat, and now I leave the floor to the next presentation of Kim Garabo.
Okay, thank you, Daniele. And yes, it's very important to have this tool available, because these impressive massive mortalities of Pinna nobilis altered our seascapes in a dramatic way. And so to have this tool now offer the possibility to record how these species will recover, and if will recover. So please, in case of doubt or curiosity, ask to to Daniele in the chat, and now I leave the floor to Kim for the last session of this webinar. Thank you, Kim. I leave your floor.
Yeah, thank you, Carlo. Thank you, Daniele and Ivan, for this thoughtful presentations. So I'm going to start my presentation, which is a topic on that I mentioned at the beginning, the sea urchins populations. So this a new protocol that we developed in the MPA Engage project, but in fact it comes from a long experience, previous experience in many areas of the Mediterranean. So it has the protocol that I'm going to show you, the way of measuring or characterizing the sea urchin populations. It's something that is robust and well known for many years now. So it's just to put it on the spot to spread its application. So probably the first question that we have to ask ourselves is why it's important to monitor sea urchins. And in this slide, I show you what I mean, nature is complex and there are a lot of interactions between the different components, the macroalgae, the sea urchins, the fishes, and other invertebrates. But just to put it in a simple way, sea urchins can play a key role as herbivorous species. So when they are abundant, they can drive the macroalgal beds to the barren. So in this in your right side of the figure, if we have fishes that predates on the sea urchins, what happens in when a marine protected area is working well, then what we have is that the macroalgal beds can stand for this rich biodiversity and providing these ecosystem services that they provide. But once if we let the fishers go there and start overfishing, then we move from these rich macroalgal beds to these barren with a lot of sea urchins and just a loss of biodiversity on the other, on not only on macroalgal but in other species. However, as to show this how the fishes act or interact with the sea urchins, I'm going to show this footage, this short video made by Jordi Boada. It's footage of a predation experiment that they conducted in one MPA. So if we can play the video. There is some sea urchins attached, and then you see the Diplodocus argus coming to bite the sea urchins, and then there are other species like the Sparus aurata and other Labridae species. So you see that when the fishes, the sea urchins are, depending on the size, but the fishes can go there and predate on the sea urchins. So when there are no fishes, this is why the sea urchins grow in their populations. So maybe we can go back to the presentation. Thank you. So this was an example of how predation can somehow regulate the sea urchins populations. But what is true and is that there is the climate change, so the warming that we have been seeing, and you will have more information if you watch one of the webinars that we provided this week. So we know that the Mediterranean is warming, and along the warming, we know that some other key species like the herbivorous fish, the salpa, the salema, is affected by the warming. So the populations of these species are decreasing. So this can have some impacts as well in the macroalgal beds. But we have now as well the rise of the arrival of the colonization by exotic species like the rabbit fish, the Seagrass species, that they are herbivorous fishes coming from the Red Sea, and they can as well play a role on this. And we know as well that the most common sea urchin, the Paracentrotus lividus sea urchin, is really it can be affected by the increase of temperature. In fact, there are some late works studies on the thermotolerance of this species, and some evidences that in the Eastern Mediterranean, in the coast of Israel, this species has experienced even local extinctions. So we have the disappearance, the decrease, and even the complete absence of these species. And we know that there are other species here. I put this red, these black sea urchins to identify changes in the sea urchins species that can be present in the areas, like the black sea urchin, the Arbacia lixula. So I know this is more complex than this, but it's just to show you that the central role of the sea urchins can play in shaping the structure and the functioning of the macroalgal beds in the Mediterranean, and as well there are many other interactions. But I think that we now agree that the sea urchins play an important. It's important to monitor them. So for the monitoring, we target two main species, the Paracentrotus lividus, which is considered, and we have some evidences that is a cold affinity species, or at least when the temperature is increasing, they have problems and they suffer mortalities. And it's a species that it's a main herbivore in the macroalgal beds. And the second species is the black sea urchin, the Arbacia lixula, which has a different thermal sensitivity. It seems that it's more present in the warmer areas in the Mediterranean, and it has a different foraging behavior, and it can provide some changes in the impacts in the macroalgal beds, the way that this sea urchin is acting, because it scrubs more it can eat even the calcareous algae. So the goal of the monitoring protocol that we propose is to track changes on the populations of sea urchins. So here we have a population. So what we want to know is to quantify the density, the abundance of this species, and the size structure, because this can tell us what is going on. And here you have I put some figures from one of the many papers tracking the changes in the different areas of the Mediterranean. But you see in the upper figure changes in the density, so the mean of the individuals by the surface, and how these changes across a long time. And in the bottom plots, what you have is the size structure, so the frequency of the different sizes of the sea urchins in different years that you see here. Okay, so for instance, here you see that in this case you don't have small sizes, but in this other population you have small sizes. So who can implement? Mainly this protocol can be implemented by MPA managers and research of scientists. I put it as well trained recreational divers, because if we have experiences, so for instance with some university students that can be trained and to collect information on the sea urchins populations as well. Regarding the materials, it's not really really costly materials. We need a caliper that we can go under the water, and an underwater board with where to mark the sizes and the density of the sea urchins. Measuring tape to mark the transects that we will see in the next slide. Some globes, because you know, the sea urchins can with the spines can be tricky to play with them, and 1 meter bar, because this is the way, as we will see now, to conduct the transects. So for the field operations, what we propose is to deploy 3 50 by 1 meter transects, and we can conduct these transects between 3 and 10 meters depth, and in the ideal or the minimum sampling effort that we have to do is to work in 3 sites, just to characterize what's going on inside the marine protected area. Of course, these 3 sites, they have to share similar habitat features, just to be able to compare across the different sites. And then we start the transects and measuring the size of Paracentrotus lividus and Arbacea lixula that we find within the transects with the caliper, of course. And what we have to do is measure the size of the testes, not so without the spines. So what we propose, because in some cases the abundance of this species is really high, so we don't want you to measure hundreds and hundreds of sea urchins. So what we propose is to measure up to 100 individuals per transect, because in this way we have already a good estimation on the size structure of the populations. It can happen because we have examples of this, that the sea urchins suffer as well some mass mortalities, in some cases linked to some diseases. So this is something that we can as well quantify in the while we are doing the monitoring. And of course, this information can be included as in the assessment of mortality in the marine protected area. So this more or less how once we go to one place, and this is the template that we provide to go into the field and quantify the abundance and the size classes of the different species. Once we have this, we have as well, for the other protocols, an Excel template where this data can be included, and then we can we develop this tool, as Daniele was mentioned earlier, for the Pinna nobilis protocol. We developed this tool where these Excel templates that we provide, they are uploaded to this Power BI tool. So this tool is supported by this software Power BI. This is free to use, and this is available. The tool is available as one of the materials that we provide in our website. So this can help in the data management, because the managers, they have to go through the Excel file. So they implement they put the data there, and then they can take advantage of this tool to display the data and to understand what's going on with the different protocols, and in this case with the sea urchins populations. For instance, here what is showing this figure is the map with the position of the sites that have been monitored, and then the density of the 2 species that we included. We added as well the Sphanercinus granularis, which is another sea urchin species that in some cases can be abundant. So you can add as well this species, and then the protocol can take this. And as the previous figure, it was for the abundance, and here what you have is the size structure of the populations, and as well the mean size of the population. So this, it provides, as I say, information about to characterize the sea urchins populations. And just to finalize, I want to give you one of probably the most meaningful example of the monitoring of the sea urchins that it has been conducted by in the Meadows Island by a team from the University of Barcelona that have been monitoring using the protocol that I just show you in the Meadows Island for the last 25 years. And I'm going to give you some really fast overview on the results that they obtain. Here, what you have is the density, so the changes in the density. In the orange line is outside the MPA, and the blue line is inside inside of the MPA. While the part of the MPA where fishing is not allowed, and the orange one is in the zone on the areas of the MPA that this fishing it was or is still allowed in under some conditions. So what we see here is already that there is some changes in the density. The magnitude of these changes is higher in outside of the in the fished zone because what we consider is that the fishes inside the MPA, the abundance of the predator species is higher, so they keep these populations in more stable. And if we look at the size structure in the right plots here, what you see is that inside the zone where the fishes are more abundant, we lack of these peaks of recruits, so these peaks of small sea urchins. So this gives an idea that the fishes, in fact, they regulate quite well the abundance of these species, and from this the interest of having the MPAs to provide conservation on the macroalgal beds. So if we move farther on time, we have in 2008 there was a really big storm in the Catalan coast, so affected the populations, and you see here a decrease because this big storm with big swells and waves. They kill many sea urchins, and you see that there was this decrease in the density of sea urchins in both areas, in the fished and in the fished area. And what we see, and what is interesting from these results as well, is that the recovery of the populations, it was much faster. I mean, since there was not the control by the fishes outside in the area that where fishing is allowed, there is less fishes, and then the populations rise faster. While in the non-fished area within the MPA, the population of sea urchins it's much more under control. So this is good news for the conservation of the biodiversity. And that's all from my side. Thank you very much. And I hope that if you have any comments, I'm more than welcome to answer them. Thanks.
Thank you. Thank you, Kim. Very important methodologies that we hope that can be shared and applied in different MPA across the Mediterranean Sea. Now, of course, you can put your curiosity, your question in the chat. We can start so the discussion on these protocols, and there is for the moment one question from Paul. Is a general question. It's not specific on this protocol, but is an important point, because Paul asked if it is important to have a continuous monitoring inside the MPA, or it is enough to have some spotted monitoring. And I think that, for example, what you explained, Kim, yesterday for the temperature data logger is something that can better clarify the opportunities that a continuous monitoring can offer.
Mm-hmm. Yeah, I can take this one. Yeah, having this system of buoys is it's a good system, but it's quite costly. I mean, it's we talk about hundreds of thousands of, probably tens of or hundreds of thousands of euros. So if the MPA can manage to buy these equipments, that's fine. And of course, high resolution information, it's much better than discontinuous information. So yeah, if the MPA is able to buy these big systems, that's fine. What we propose is to use this, now the technology offers us some opportunities. So for instance, for the temperature, as we show in yesterday's webinar, dedicated to the tracking of the temperature changes. We use these devices that has a cost of about 100 euros, and they provide information every hour of temperature. So we have been collecting continuous time series on hourly basis on temperature. There are now new devices like this. I just saw the other day that there is that the cost is higher, but there are similar devices for a pH, for instance, or for turbidity. But we have to keep in mind that in some cases you need some maintenance and you need some personnel dedicated to this. But yeah, definitely having some devices and monitoring these physical chemical parameters in the MPAs is fine. Thanks.
Thank you, Kim. Yes, the point is that one of the added value of these protocols is that at least the experience that we are have with in these years with the project is that there is a lot of interest also from diving center and citizen science to be part of this process. And in fact, exactly as the temperature data logger is something that in some MPA is managed by divers that support the staff of the MPA in the maintenance of this data logger. And I would like to ask to Ivan if also Posidonia oceanica, that is not so exciting in terms of habitat for divers in general, is something that can attract the interest of citizen scientists, or is a protocol that probably is more
addressed to scientists
by the staff and by scientists.
No.
What do you think?
No, actually, some MPAs in the Mediterranean, for instance, the Tavolare MPA, are developing citizen science approaches to involve divers, and Posidonia oceanica is one of the protocols and one of the species that is monitored by divers. The experience of MPA Engage in the last 3 years suggests us to that this protocol is can be applied by divers, but training and intercalibration is needed, because counting Posidonia shoots is easy, but people have to be really interested and do this with precision and accuracy. Otherwise, we get not useful data, and generally wrong data can provide bad information about the condition of the meadows, and so it's very important to have trained and training and intercalibration processes very effective. If you are able, MPA are able to do this, for sure, also divers are able to provide useful data.
Great. Thank you. So I think that also citizen science can be engaged in this kind of activities. And another point, always thinking about the opportunity to be helped by citizen scientists, for example, is that another opportunity that this protocol offer is that they are easy, no, in terms of application. And it is possible sometimes to merge, different protocol during the same dive. For example, there are MPA that they start, for example, with the assessment of the massive mortalities in deeper water on gorgonians. Then they can do the visual census of fish, in the upper layer. And so, optimizing the time dedicated to this protocol. And I wonder if, for example, also sea urchins and Pinna nobilis is something that can be merged. I don't know if Daniele can probably have already some experience on this.
Yeah. Of course, Pinna nobilis is since it is an iconic species, it's a bit more attractive than the others. And what we saw from previous experience is that there is a big feedback on the citizens. Of course, you have to be careful, explain everything. For example, regarding the alive specimen that, for example, can be recognized because of stands. Sometimes they reported us, of course, that the individual is dead, but if you explain carefully that you have to go close, to have to see the mantle and the shells that is closing, and if you intercalibrate all this information and provide all this information, explain the context, the feedback is very high. And I think it's also true for Posidonia oceanica, like was commenting the last comment of the chat. If you explain the background, the citizens will be completely involved and be part of the research. It's of course very, very important. And another point is about the training and the calibration specific for Pinna is that now, for example, some recruits have been found. They are rare, but still there are some recruits. So it's extremely important that citizens will be trained to be careful and don't damage the fragile individuals, of course. So this is a very important point. That's why we are here. That's why we are explaining the protocols and all this important information.
Okay, thank you, Daniele. Yes, we have also Tatiana that is ready to apply another protocol this summer with the diver Tenistica, we hope. And yes, I think that probably now Kim, as coordinator, I don't know if you want just to say something after this series of webinar. I think we are doing a very great job. I'm receiving a lot of feedback from divers that are already applying this protocol. I think that this system of protocol is offering a very great and unique opportunity for the first time to share methodologies and start actually to work together, in the Mediterranean Sea with the same approach, with the same vision. I think it is a very important moment and important results, but I prefer if you, Kim, that I put a lot of effort on this.
No.
Can probably say a couple of words because I think we are reaching very good results.
Thank you, Carlo. You are part of this process as well as Daniele, and Ivan, and all other speakers that have been participating in this webinar series. I'm really proud to see all the work that we achieve, and what I just want to stress is that the protocols that we are providing here are guided by this philosophy of cost effectiveness. So this is something that we work a lot on, trying to get as much as information possible with the good resolution at the lower cost, with this vision of having this regional perspective, so that it can be implemented everywhere to get a response about the local responses, but with this view on the regional and the global impacts of the climate change and other impacts that are happening or that are impacting the Mediterranean. And the third pillar of these protocols behind the development of this protocol is the engagement of the different actors, the different stakeholders. So the managers, of course, the scientists, and the recreational diver, the fishermen. And I think that this is a way of getting and expanding our capacity of gathering information. So expanding, upscaling our scales of observation, both in time and in space, but as well raising the awareness on the impacts that are happening, and taking adopting the solutions and learning what is working in some places and in others. So for me, this is not only gathering information, it goes beyond. It's providing the basis to work or to contribute in or to take action for the solution. So I think that is the major outcome of this. I just want to say that all the documents, the materials are available in the website, and you are more than welcome to visit there and check the materials and all the video tutorials, all the video materials that we have, the presentations, the documents. And we are now, by the end of the project, we will be finalizing a new booklet with the protocols. So the 11 protocols that have been developed and tested during the MPA Engage and refined thanks to the feedback from the implementation in the different MPAs participating in the MPA Engage project. We will be delivering this booklet that it will be as well available in the website, and you are more than welcome to have it. So thank you very much for your interest in this. And as Carlo say, we have to work together. It's the only way to find effective solutions to this big challenge that we have ahead with the climate change and with the biodiversity loss crisis. Thank you very much.
Thank you.
Thank you.
And see you soon. Bye-bye.
See you. Bye.
Bye.
Bye. Bye.